Dear students, myself Dr. Zubaria Arham Javed, Faculty of Botany Department of Microbiology subject BOTN 4128. Today we will discuss about lecture number 27, which is from Control of Microorganisms, your course outline, and which today we will discuss about the use of physical methods in microbial growth control, and it's the second portion of physical methods, that is radiation. Here is the contents. Like, first of all, we study about the radiations, then we study different types of radiations like non-ionizing radiation, ionizing radiation, and microwave radiation. Let's look on our previous lectures. As you all know that in our lecture number 25 we discussed about the physical agents like heat, dry heat, and moist heat, that how heat controls the microbial death. And then in our lecture number 26 we discuss about the mechanical removal methods like filtration of air and filtration of liquids. And today in our lecture number 27 we will cover the topic of radiation and physical agents, in which we study about the ionizing radiation including X-rays, cathode, gamma rays, and the non-ionizing radiation including the UV rays. Dear students, I again repeat the terminologies that you must know, like disinfection, the destruction or removal of vegetative pathogens, but not bacterial endospores, usually used only on inanimate objects. Then sterilization, the complete removal or destruction of all viable microorganisms that just used on inanimate objects. Antisepsis, like chemicals applied to body surfaces to destroy or inhibit the vegetative pathogens. And last one is chemotherapy, like chemicals used internally to kill or inhibit growth of microorganisms within the host tissues. You all must have the idea about these terminologies. Today we will discuss about the physical methods like radiations, and you all have idea that there are 4 types of radiation. Means non-ionizing radiation, ionizing radiation, microwave radiation, and the strong visible light that emits from the sun mostly. And you all know that non-ionizing radiations include UV rays like ultraviolet light, and the ionizing radiations must include X-rays, gamma rays, and cathode rays. Here you see a graphical presentation of non-ionizing radiation and the ionizing radiation. Here is the partition between non-ionizing and ionizing radiation, and it's clearly see that ultraviolet, X-rays, and the gamma rays are included in the ionizing radiation, and the radio waves like low frequency, extremely low frequency rays, and the microwaves are included in the non-ionizing radiations. and it also includes the infrared and the visible light. Let's start. First of all, non-ionizing radiation, which includes ultraviolet light. And today we will cover the introduction, the wavelength range in which the ultraviolet light works, then the mechanism of action of ultraviolet light, then how ultraviolet light works, then what is the DNA repair mechanism of bacteria. Then we will see the lethal and effective range in which ultraviolet light gives their best result or effectiveness. Then water treatment, that how UV rays or UV radiations work to sterilize the water, to sterilize water or is helpful in the water treatment. Then the microbial sensitivity to UV radiations, then their applications, and in the last we will discuss about the precautions. As you all know that microbiologists take advantage of the effects of ultraviolet and non-ionizing radiation to sterilize or disinfect objects. Ultraviolet light consists of light of wavelengths between 40 and 390 nanometer. But wavelengths in the range of 200 nanometer are most effective in killing microorganisms by damaging DNA and proteins. When we see about the mechanism of action, then ultraviolet light is absorbed by the purine and the pyrimidine bases of nucleic acids, and such absorption permanently destroys these important molecules. Ultraviolet light is especially effective in inactivating viruses. However, it kills far fewer bacteria than one might expect because of the DNA repair mechanisms. Once DNA is repaired, new molecules of RNA and protein can be synthesized to replace the damaged molecules. When we discuss about the lethal range of ultraviolet rays, unlike ionizing radiation, the energy of UV radiation is lower and its power of penetration is poor, while UV radiation around 260 nanometer is quite lethal, but does not penetrate the glass, dirt films, water, and other substances very effectively. Because of this disadvantage, UV radiation is used as a sterilizing agent only in a few specific situations. Lying in soil and exposed to sunlight for decades, endospores are resistant to UV damage because of a small protein that binds to their DNA. This changes the geometry of the DNA by untwisting it slightly, thereby making it more resistant to the effects of UV irradiations. Here is a graphical presentation which shows that ultraviolet radiation, which is further divided into ultraviolet C rays, ultraviolet beta rays, ultraviolet alpha rays. These are in the range of 100 to 400 nanometer, and the visible light is having the wavelength range between 400 to 700 nanometer, and the infrared rays have more than 700 nanometer wavelength. It does penetrate the air, effectively reducing the number of airborne microorganisms and killing them on surfaces, in operating rooms, and rooms that will contain caged animals. Ultraviolet lights lose effectiveness over time and should be monitored often to help sanitize the air without irradiating humans. UV lights can be turned on when the rooms are not in use. We must take care about that UV lights must be turned off when we are using the room or when we are using the working place where the UV lamps are placed. Ultraviolet radiation can be produced artificially by mercury vapor lamps. Here is the graphical presentation which shows that how UV ultraviolet C rays blocked by atmosphere, and it is most dangerous for all living organisms. And the ultraviolet beta rays which causes skin to dark and also causes skin cancer on longer exposure, and it also reaches the lens. UV ultraviolet alpha rays also reaches the lens and retina of our eye and also cause cataracts and retinal diseases. Exposure to ultraviolet light can cause burns, as anyone who has had a sunburned nose, and can also damage the eyes. Years of skin exposure can lead to the skin cancer. As you see from this figure, that how sun emit ultraviolet beta rays and ultraviolet alpha rays, and ultraviolet beta rays affects the dermis layer of our skin, and ultraviolet alpha rays damage or affect the hypodermis layer of our skin. Here you see the UV lamps situated in the labs. UV lamps are sometimes placed on the ceilings of the rooms or in the biological safety cabinets to sterilize the air and any exposed surfaces. Here the UV lamps are placed on the ceiling of the rooms. This is the more clear picture of UV lamps that are placed horizontally. These are easily available in our superstores, in our utility stores, in labs, in hospitals, in industries. Okay, here is the commercial ultraviolet units which are available for the water treatment. When water flows through racks of the UV lamps and is exposed for 254 nanometer UV radiation, the system has a capacity of several million gallons per day and can be used as an alternative to chlorination. Pathogens and other microorganisms are destroyed when a thin layer of water is passed under the lamps, and it is the most commercial method used for the water treatment or to sterilize the water or to remove the microbial organisms from the water. When we discuss about the microbial sensitivity to UV radiation, then we must know that bacterial spores are generally more resistant to ultraviolet light than are vegetative cells. And viruses are also inactivated and tend to be more sensitive than bacterial spores. Human immunodeficiency virus is not inactivated by ultraviolet radiation. Human immunodeficiency virus, as we all commonly call it the HIV virus, is not inactivated by UV radiation. When we discuss about the applications of ultraviolet radiation, we must know that it is very effective to disinfect the drinking water, as we already discussed in water treatment portion, and it also effective in disinfection of enclosed areas such as entryways, hospital wards, operating theaters, laboratories, and in ventilated safety cabinets in which dangerous microorganisms are being handled. And whereas the risk of using ultraviolet radiations, we must avail some precautions, because UV radiations burns skin and damages eye, as we already discussed in previous slides, that how UV radiation affect the eyes and causes cataracts and retinal damages, and how it causes skin cancer. So people must working in such areas must be certain that the UV lamps are off when the areas are in use. Second portion includes the ionizing radiation, in which we will cover the introduction about the X-rays and gamma rays, then radiation unit, commonly called RADS, then mechanism of action of ionizing radiation, then some of its applications, and then cold sterilization. But what is cold sterilization, and why we call the ionizing radiation a cold sterilization? Here you see a symbol. This is the universal symbol for irradiation that must be affixed to all irradiated materials. X-rays, which have wavelengths 2.1 to 40 nanometer, and gamma rays, which have even shorter wavelengths, are forms of ionizing radiation. So named because it can dislodge electrons from the atoms and creating ions. Means longer wavelengths are forms of non-ionizing radiations. These forms of radiation also kill microorganisms and viruses. Many bacteria are killed by absorbing 0.3 to 0.4 millirads of radiation. As for example, polioviruses are inactivated by absorbing 3.8 millirads. Here the rads is a unit of the radiation energy absorbed per gram of tissue. A millirad is 1/1000th of a rad. Ionizing radiation is an excellent sterilizing agent and penetrates deep into objects. It will destroy bacterial spores and vegetative cells, both of prokaryotic and the eukaryotic microorganisms. However, ionizing radiation is not always effective against the viruses. When we discuss about the mechanism of action, then ionizing radiation damages the DNA and produces peroxides, which act as a powerful oxidizing agents in cells. This radiation can also kill or cause mutations in human cells also if it reaches them. Here you see an irradiation machine that usually uses the radioactive cobalt 60 element as a gamma radiation source to sterilize fruits, vegetables, meat, fish, and spices. So class, we will end our today we end our today's lecture on the mechanism of action of ionizing radiation, about applications and the precautions. We move on to the next lecture. Here are some references about today's lecture. These are the references of some textbooks like from Surinder Kumar and the Microbial control flashcards, and this is the microbial rate of microbial death, which is a Google link, and this is your reference book of K-Line's Microbiology. It's the book of microbiology from Prescott and Hurley. Okay class, if you have any query relevant to today's lecture, then you are freely ask me in Google Classroom. Thank you so much.